Preliminary exam name, Amira Kefi Advisor, Dr. Chunyu Liu. Title, Improving Human Genome Annotation by Detecting and Validating Novel Coding and Non-Coding Genes/Exons/Isoforms in the Human Brain Using Integrative Analysis. The human genome annotation is still far from being complete, Abascal, F. et al., 2018. Especially in terms of non-coding RNAs, regulatory sequences, and alternatively spliced transcripts, Mudge, J. M. et al. 2016. A complete list of functional regions of the reference genome is very crucial for biomedical studies and medical screenings, Frankish, A. et al., 2015. In fact, the gene annotations are extensively used as reference in all Omics studies, Zhao, S. et al. 2015. However, in studies using microarrays, probes are limited to the annotation file list of genes. In studies using whole genome sequencing data, like whole exome sequencing projects and genome-wide association studies, GWAS, analytical procedures will discard the mapped reads to the unknown regions such as intronic or intergenic regions. Therefore, this can have a dramatic effect on both gene quantification and differential analysis, Zhao, S. et al. 2015. Several reasons might explain why different genes failed to be annotated, Cassandra, 2018. The first reason could be related to the spatiotemporal nature of those genes that are associated with the different cell tissues or developmental stages, Namuva et al., 2013. A second reason could be related to a phenotype. For example, certain genes might be only expressed at a specific disease state or under a specific treatment. Another important reason for the missing annotation is related to the sequencing technologies, or to pipelines pre-processing steps or even to downstream analysis tools. In fact, due to the elimination of low abundance and duplicated regions, which is a standard step in next-generation sequencing pipelines, many genes that share the same reads are not detected. Therefore, when a gene is quite long with complex splicing, many isoforms could be missed by reads from short-read sequencing, RNAs, because they can only span one or two junctions. Consequently, in order to fill the gap, hybrid sequencing combining RNAs with IsoSeq is currently the most effective technique for the discovery of novel genes and isoforms. We hypothesize that the third generation of sequencing using long reads enables us to find novel genes/exons/transcripts that cannot be detected by the second generation alone. Adopting an integrative analysis approach of different generations of sequencing can allow us to confirm and subsequently validate the novel genes/exons/isoforms. Aim 1. To detect novel genes/exons/isoforms using third generations of sequencing and confirm using second generation of sequencing. We perform a systematic search for the novel exons, transcripts and genes in human brain tissue using a PacBio IsoSeq. We combine second and third generation sequencing of RNA from a normal brain tissue sample to identify novel genes, exons and isoforms. First, we confirm their transcription using RNAs data. Then, in order to confirm a subset of putative coding genes, we used RiboSeq and mass spec data to verify the novel genes. Aim 2. To computationally classify novel isoforms/exons/genes into coding and non-coding using Bayesian inference. We hypothesize that we can use different omics data and sequence features to perform a classification of novel genes in different types of coding and long non-coding genes. Therefore, we obtained the GeneCode long non-coding and coding genes and performed feature selection through PCA analysis to assess the most discriminative features. The sequence feature selected included the probability of coding, the Fickett score and the hexamer score of each gene. The Omics features includes the RNAs score and the RiboSeq score. Based on the known genes from the GeneCode annotation, we used Bayesian inference to estimate the parameters of the multiple regression model that can be used to classify the novel genes. Aim 3. To identify novel isoforms/exons/genes that may contribute to neuropsychiatric disorders. We hypothesize that some of the novel's isoforms/exons/genes are involved in neuropsychiatric disorders. We will inspect the DBSNP, ClinVar, GWAS catalog databases and investigate whether the novel isoforms/exons/genes are covering any SNPs that are relevant to neuropsychiatric disorders. This project will lead to the development of a novel approach and method to detect and validate novel isoforms/exons/genes that can be used to improve the quantity and quality of annotated elements in the human genome annotation. Furthermore, this work will also provide a fundamental investigation and understanding of the challenges and the reasons behind missing isoforms/exons/genes in the human genome annotation. Research plan. Significance. So far, the genome sequencing revolution has led to the sequencing and assembly of many thousands of new genomes. However, genome annotation still uses nearly the same technology that we have used for the past 2 decades. The complete construction of genomes needs the use of fully automated procedures for annotation, but errors in annotation and missing elements, Frankish, A. et al. 2015 are still as prevalent as they were in the past, Abascal, F. et al. 2018. As a result, in the last 5 years, hybrid sequencing combining RNA-seq with iso-seq has been accepted as the most effective technique for the discovery of novel genes and isoforms. Therefore, in this work, we performed a systematic search for the novel isoforms/exons/genes in human brain tissue by combining second and third generation sequencing of normal brain tissue samples. Challenges in novel gene discoveries and validation were investigated in recent efforts toward the completion of the human genome, Pertio, M. et al., 2018. The gene finding and validation problem in the human genome is still difficult and challenging, Salzburg, S.L., 2019. So far, only about 1.3% of the human genome comprises protein-coding exons. In fact, in a recent comparison of all the protein-coding and L-Chunyu transcripts in the RefSeq and GenCode human gene databases, only 27.5% of the GenCode transcripts had exactly the same introns as the corresponding RefSeq genes, Pertio, M. et al., 2018. Thus, even after 18 years of effort, the precise exon-intron structure of many human protein-coding genes is not settled. The annotation still suffers because the best automated gene finders are far less accurate and manual curation is always needed. However, manual curation and validation is still prone to errors and time-consuming. Therefore, a more sophisticated and automated process using the cutting-edge sequencing techniques needs to be established. In this project, we adopted an integrative analysis approach using different generations and types of sequencing to detect and confirm the transcription and translation of novel isoforms/exons/genes. However, the definition of non-coding genes is also a challenge, leaving a large amount of misclassification. Therefore, we will develop a probabilistic framework to classify the non-coding novel genes by integrating different Omics signals and sequence characteristics. 2. Innovation. An integrative framework and pipeline to improve the detection of novel isoforms/exons/genes and confirm their transcription and translation signal. So far, RNA-seq has been the gold standard technique, Pertio, M. et al. 2018, producing high-quality and high-throughput reads of the sequence. Nonetheless, due to RNA-seq short reads, different tools, Jaraborg et al. 1999, have been used to predict splice junctions but still face challenges to reconstruct the splicing model in a continuous way to obtain a complete transcript. This major limitation hampers the ability of RNA-seq to identify novel isoforms. The emergence of third-generation sequencing, IS-ASEQ, from Pacific Bioscience, PacBio, or Oxford Nanopore, provided significantly longer reads, with the length up to 7 kilobits. However, some of the long reads had lower raw accuracy and sequencing depth. As a result, hybrid sequencing and/or analysis combining RNA-seq with IS-ASEQ are currently the most effective techniques for the discovery of novel genes and isoforms, OKF et al., 2013. In this project we are integrating different Omics data from third generation and second generation of sequencing to detect the novel isoforms/exons/genes with high confidence for transcription and translation. Therefore we will detect the novel isoforms/exons/genes using iso-seq data, then we confirm the transcription using RNA-seq data. Next, we investigate the translation using RiboSeq and mass spec data. Figure 1. Workflow used for the identification and confirmation of novel isoforms/exons/genes. A computational validation using a probabilistic framework that integrates the different omics and sequence features to classify novel isoforms/exons/genes. There are different types of data integration depending on the purpose and on different types of omics data and different machine learning methods. Wu, C, Zhao, et al., 2019. However, the first important step is to perform feature selection for our integrative analysis, which means the most important genes characteristics that can help us classify the novel genes in the appropriate category of the human annotation. We have many types of omics: RNA-seq, riboseq, ADAC-seq, mass-spec. The important question is how to find which subset is important in all of them. Choosing the subset of the most important and right features does improve integration performance and accuracy as shown by Lei Cao, K.A. et al. 2009. For example, using dimension reduction and clustering analysis are very important factors to select the best features. Meng et al. 2016, Fang, Z, et al. 2018. Therefore, we used principal component analysis to select the most discriminative features between the coding and non-coding genes in the human genome gene code annotation. Next, we used Bayesian inference to estimate the parameter of these features in multiple regression analysis. Ultimately, the parameters estimations will be used as prior knowledge to classify novel genes in the coding or non-coding categories of the genome annotation. 3. Approach. 3.1 An integrative approach for detection and confirmation. Aim 1. To detect novel genes/exons/isoforms using third generations of sequencing and confirm transcription and translation using second generation of sequencing. A. Preliminary results. We used 3 IS-OSOC samples. One control sample, CSU China South Central University, was isolated from the nuclei of post-mortem cerebellar tissue, and 2 public samples from PacBio repository, a control sample, PB normal, and an Alzheimer's sample, PBAD, also from brain tissue. In addition, we had 288 RNA-seq samples and 197 riboseq samples of human brain tissues that were obtained from controls, bipolar disorder, and schizophrenia brain tissue samples from the PsycEncode project, Akbarian, S. et al., 2015. Novel isoforms/genes. We selected the Sconti algorithm, Tartaglia et al. 2017, for our analysis. We first collapsed compatible reads to isoforms. We then compared each isoform to the previously annotated exon intron structure to detect different alternative splicing, AS, events from the results from the brain tissue sample. The Sconti tool detects, quantifies, and predicts different isoform categories using splicing types along novel isoforms as well as known isoforms inferred from iso-seq reads. First, the known transcripts were detected. A known transcript can be classified as a full splice match, FSM, if it matches a known transcript at all splice junctions. However, it is an incomplete splice match, ISM, if it matches consecutively, but not all splice junctions of a known transcript. As for the FSM transcripts, we found 28.3% in the CSU sample, 58.9% in the PB normal sample, and 46.5% in the PBAD sample. Although, when we restricted the transcript length to the 1 to 2 kilobits, the percentages dropped to 4.8%, 6.9% and 21% respectively. For the ISM transcripts, we obtained 23.7% of the transcripts in our CSU normal sample, 17.2% in PB normal sample, and 23.1% in PBAD sample. This then decreased to 11.2%, 10.3%, and 4.6% respectively when we considered only the 1 to 2 kilobits transcript. Other transcripts had also been detected as subcategories of FSM and ISM, like UTR3 fragments, monoexonic and multiexonic transcripts. Table 1. Long reads characterization from CSU iso-seq sample. Novel exons. We performed mapping of the long reads and used an in-house script to compare the exons detected by iso-seq with the most used annotations, GenCode, RefSeq, and Chess, Pertio, M. et al., 2018. The IsoSoc data detected 221,080 exons. Among them are 14,370 novel exons that are distributed proportionally to the chromosomes. Next, we wanted to know how many of these exons could have transcriptional signals from short-read sequencing. For this reason, we used 188 RNAs samples from brain tissues available at the PsycEncode project to confirm the transcription. We also used RiboSeq profiling data of 194 brain tissue samples from the same repository to detect the translational signal. From the 14,370 novel exons, we found mean expression RPKMs equals 36.280 and median expression RPKMs equals 9.804, while for RiboSeq mean expression RPKMs equals 1.174 and median equals 0.039. However, for the known exons, the mean expression RPKMs equals 11.62 and median expression RPKMs equals 0.0. While for RiboSeq mean expression, RPKMs equals 27.75 and median equals 0.0. Almost 90% of the novel exons have non-zero transcriptional signals, of which there's 70% that have translational signals in RiboSeq data. However, for the not novel exon we have 21% that have transcriptional signals in RNAs and about 91% of them have the translational signal in RiboSeq data, which suggests that most undiscovered exons are non-coding. We wanted to find out how the transcriptional and translational signals are distributed in the novel exons compared to the known ones. In our study, we found that most of the novel exons are more likely to have much lower transcription and translation signal compared to the known one. This low expression could explain why many exons are being missed by short-read transcript reconstruction tools that filter them out as noise. The length distribution of novel exons and known exons looks similar in general. However, if we take the exons with length less than 500 nt, we notice a totally different distribution showing that the missing exons are smaller ones, less than equals 100, that could have been considered as transcriptional noise or thrown away because of cutoffs used by transcript reconstruction tools. We also found that the majority of novel exons unexpectedly have 5 times the transcriptional signal than the translational signal, log2 FC equals 5, while the majority of known exons have similar transcriptional and translational signals. Finally, we observed that most novel exons are the first or second exons in the transcripts, which can be explained by the bias in the 5UTR region that is a common problem in the short-read sequencing and detection methods. Novel monoexonic genes, MNEG. Many approaches had been adopted by different annotations to find and validate novel genes. However, we need to agree on the definition of a gene and its characteristics. In the past few decades, the definition of gene has evolved from a single segment to a combination of many segments, exons. In addition, the structure complexity and the regulatory elements dictionary of genes have also increased drastically, promoters, enhancers, etc. Moreover, the percentage of protein-coding genes versus non-coding ones has changed a lot since we first thought that most genes are protein-coding, but now more evidence showing that the non-coding genes are more abundant. In this work, we report 2 major classes of novel genes, multi-exonic genes and monoexonic genes. We are interested first in the NAIG, which has not been studied and has been discarded in many studies, OKF, et al., 2013, Pertio, M. et al., 2018. New genes are generally defined as located in the intergenic region. However, the majority of monoexonic genes detected in our PacBio sample CSU were intronic. We detected 988 MED where 845 are novel. We observed that the number of novel MEGs are not proportional across the chromosomes and that they are more abundant than what we already have in annotations. This suggests that this type of genes had probably been ignored due to different computational and biological reasons. Furthermore, we investigated the transcriptional and translational signal of the novel MEG, and found that the median transcription is 10.8200 and mean transcription is 25.5700, while for the translation we have median equals 0.07704 and mean equals 0.40470. However, for the known MEG we found median equals 18.0900 and mean equals 53.1600 for transcription, while for the translation the median equals 0.17900 and mean equals 0.83240. We wanted to know if the monoexonic genes were only a specific event in our PacBio sample, so we used two other samples from PacBio repository. PB normal is a normal brain sample and PBAD is an Alzheimer brain sample. New genes are generally defined as located in the intergenic region, and most of the novel genes had a single exon for the 3is OSUC data. We found that the PB normal sample and PBAD sample have the highest percentage of monoexonic fragments in the ISM category, with 54.4% and 63.1% respectively from the total number of the monoexon fragments. However, in our normal sample the highest monoexonic subcategory was genic intron, which accounted for 50% of all monoexon fragments. Our CSU sample had almost equal number of transcripts as monoexon and multi-exon due to the size selection, while the PB normal sample had about 80% of transcripts as multi-exons and the PBAD had 75% as multi-exons. B. Research plan. My plan for this aim is to include more samples from RNAs, RisoSeq, and mass spec data and submit the manuscript. I also plan to use new available datasets that have the same type of Omics datasets to confirm the efficiency of our workflow in detecting and confirming high-quality novel isoforms/exons/genes. 3.2 A probabilistic approach for classification, validation, and implication of Mudge. Aim 2. To perform computational validation through classification of novel exons/isoforms/genes using Bayesian inference. Preliminary results. We adopted an innovative approach by integrating the Omics data in addition to the sequence features to classify our novel monoexonic genes with the ln or coding RNAs transcripts from the GeneCode annotation. We also used Bayesian inference to estimate the specific multiple linear regression parameters of the known long non-coding genes from the annotation and use them for more accurate classification of the novel monoexonic genes. Bayesian modeling has two major advantages over frequentist analysis with linear mixed models. First, information based on preceding results can be incorporated using different priors. Second, complex models with a large number of random variance components can be fit. Levine, M. 1999. Feature selection. Long non-coding RNAs, ln, are one of the most poorly understood but also the most common RNA species, are those non-coding transcripts with length more than 200 nucleotides. Initially, people classified non-coding RNA, nRNA, genes as junk gene or transcriptional noise, A.F. Palazzo et al., 2015. Nonetheless, researchers found that about 70% of the genome is transcribed in various contexts and cell types, Y. Okazaki, et al., 2002, S. Jabali, et al., 2012. During the last 10 years, many efforts on ln identification have been made and many approaches have been developed to make a more accurate discrimination. Several studies, C. Wang, et al. 2013, D. Veneziano et al. 2015, have summarized and reviewed the approaches of ln identification and analysis, but a few report the discussion of ln prediction methods. Wang et al. C. Wang, et al. 2013, discussed several nRNA detection methods based on homology information and common features. Han et al. 2016 summarized the popular algorithms of ln identification and comprehensive analyses of these tools used features including coding, potential, and sequence features, Wang, L. et al., 2013. Therefore, we selected two features based on gene sequence and two features based on omics data. Based on the sequence structure, we used the Fickett score to characterize the content of the bases and their position. Fickett, J.W., 1982, Wang, L. et al., 2013. The Fickett score is independent of the ORF and distinguishes protein coding RNA and nRNA according to the combinational effect of nucleotide composition and codon usage bias. The percentage composition of each base is also determined. These 8 values are then converted into probabilities, P of coding using a lookup table provided in Fickett, J.W., 1982. First it computes 4 position values and 4 composition values, nucleotide content, from the DNA sequence. For example, position value of A, APOS, is calculated as follows. A1 equals number of as in position 0, 3, 6, A2 equals number of as in position 1, 4, 7, A3 equals number of as in position 2, 5, 8. Opos equals max A1, A2, A3 min A1, A2, A3 plus 1. Next, each probability is multiplied by a weight, W, for the respective base, where the value of W reflects the percentage of time each parameter alone successfully predicts coding or non-coding function for the sequences of known functions. Finally, the Fickett score is calculated as follows. Fickett score equals I equals 18 YP. We also use the hexamer score to characterize the coding potential of the sequence. Hexamer score determines the relative degree of hexamer usage bias in a particular sequence. Fickett, J.W. et al. 1992. Positive values indicate a coding sequence, whereas negative values indicate a non-coding sequence. This score is usually considered as the most discriminating feature because of the dependence between adjacent amino acids in proteins. Fickett, J.W. et al. 1992. For a given DNA sequence, the probability of the sequence under the model of coding DNA and under the model of non-coding DNA, and then the logarithm of the ratio of these probabilities, is considered the score of coding potential. Wang, L. et al. 2013. Therefore, for a given hexamer sequence S equals H1, H2, we obtain hexamer score equals 1 mile equals 1 m leg F high, where FFI and FFI represent in-frame hexamer frequency, calculated from coding and non-coding training datasets. Next we use the RN-ASOC and RiboSeq scores-based cufflinks quantification, Trapnell, C. et al. 2012, and a rescaling procedure as the Omics features. Our PCA analysis showed that these features are good enough to discriminate between coding and non-coding transcripts using GenCode transcripts and help classify the novel monoexonic genes. Figure 2. Feature selection of GenCode genes using PCA analysis. Figure 3. Feature selection of novel monoexonic genes and GenCode genes using PCA analysis. Bayesian inference to estimate the regression parameters. We use the Bayes multiple regression model to infer the parameters of linear regression coefficients of known genes in order to use them to classify novel genes. Sine, MS, 2014. Bayesian learning is the solution for cases where probabilistic modeling is more convenient and traditional machine learning techniques fail to provide state-of-the-art solutions. There are many advantages for using Bayes inference. First, it incorporates prior knowledge or beliefs with the observed data to determine the final posterior probability. New observations or evidence can incrementally improve the estimated posterior probability. It has also the ability to express uncertainty in predictions. One of the major challenges in machine learning is to prevent the model from overfitting. Especially when we have small data sets, the model will only learn the specific configurations of the given data that does not generalize to the unseen data. However, if we have beliefs that were gained through past experience, in our case knowledge from the annotation file, those could be used to generalize our models in the absence of a sufficiently large dataset. Therefore, with Bayesian learning, we can use such prior knowledge or beliefs to provide additional information to the models. The Bayesian framework works as follows. First, we assume a prior distribution of the parameters that does not take the observed data into account, for example p, and then we generate an initial estimate of the parameter using the prior distribution. Next, we compute the likelihood of parameters, L, observed data vertical bar. Finally, we have to draw the posterior distribution of the parameters p, theta vertical bar observed data. The posterior density is specified in a comprehensive probabilistic way with a conditional function, QD, of the parameters given the data. Therefore, we need to determine what we are conditioning on, meaning the data D, and what are the parameters Q, and consequently what the Markov chain is going to explore. We use a general linear model to represent our variates Y and covariates X. D greater than Y, X. We want to know what would the variates look like if we already know the covariates and their distribution. In other words, what is the inherent stochastic relationship between both of them by determining a probabilistic model so that in the future when we obtain new covariates we can predict the variates. The generalized regression models the statistical relationship between the variates and covariates as follow. Equals pi x, theta pi. Where the likelihood function on the left side of y and x given the parameters is decomposed into the product of the likelihood of y given both x and parameters, and the likelihood of x given the parameters. We assume that we have no selection bias and consequently the covariates are independent of the model parameters, then we can write pi equals pi x. Therefore we deduce that equals pi x, theta pi x theta. Now we need to decide what kind of form to choose for this regression. We chose to restrict our covariates to a single effective parameter through a deterministic mapping using a function f and a normal distribution n as follow. X, theta equals nu fx, theta, sigma. Where our covariates only affects but the noise is homogeneous across all the covariates sigma. In more simplistic way we have fx, alpha, beta equals beta. X plus alpha, but here we have multiple covariates and multiple slopes, therefore we encapsulate them in a design matrix as follow. Fx, alpha, beta equals n, I zini plus alpha. Fx, alpha, beta equals xt beta plus alpha. Therefore in the context of the Gaussian measurement model we obtain a Gaussian linear model as follow. X, alpha, beta, sigma equals nxt beta plus alpha, sigma. We can further inspect these estimates by looking at the trace plots and the posterior distributions of each covariate. One way to determine if the two variables are useful predictors is to inspect the location of the 95% probability intervals. If both interval ends do not cover 0, this indicates that the covariates are helpful in the prediction model. Our preliminary results on using the Bayesian estimation showed that the riboseq and RNAs scores are not good predictors since their parameter estimate contains 0. Table 2, Parameters estimates and credible intervals for each predictor. Figure 4, Posterior distribution of each parameter. We use the Markov chain Monte Carlo, MCMC, algorithm to fit models in our Bayesian framework since we have to estimate many parameters at the same time. Valderrama Bahamondes, GI. 2019. In statistics, Markov chain Monte Carlo, MCMC, methods, Van Ravenswij, D. 2018, comprise a class of algorithms for sampling from a probability distribution. By constructing a Markov chain that has the desired distribution as its equilibrium distribution, one can obtain a sample of the desired distribution by recording states from the chain. To obtain valid inferences from the posterior draws from the MCMC simulation, one should assess convergence of the MCMC chain. The investigation of the MCMC chains shows that the parameters estimation of riboseq beta 2 and RNAs beta 3 score are not converging well, while the sequence features Fickett score, beta 4, and Heeksmer score, beta 5, don't have the hairy caterpillar look, which shows that the chains did not visit enough sampling space. Therefore, we need to increase the burn-in period in the model iterations. Figure 5. Trace plot for inspection of MCMC chains convergence. Research plan. My future plan is to incorporate more omics features and more genes to improve the estimation of the Bayesian model. Next, I will use the estimated parameters as informed priors to classify the novel genes using a logistic Bayesian regression. In addition to monoexonic genes, I plan to classify the exons and isoforms. Finally, to improve the Bayes model I will increase the number of chains, iterations and the burning period. Moreover, the Gaussian noise is critical and might improve the estimation if we define with a half-Cauchy function, Gauche, J. 2018, which has heavier tail and is less constraining than the uniform given that we don't have much information about where the noise is coming from. Equals half-Cauchy 0, tau. Finally, I will establish a simulation model to assess the accuracy, precision and recall of the classification model. Aim 3. To predict the implication of novel isoforms/exons/genes in human psychiatric diseases. Preliminary results. By investigating the DBSNP database for the top 10% highly expressed novel exons, we found that 22 novel exons covered different SNPs or variants. These exons are located in the intronic region of genes and that are expressed in the brain. 4 of them were validated by PCR. 7 have biased expression in brain, 3 have ubiquitous expression, 2 broad expression, and the rest is low expression. However, none of these variants is related to neuropsychiatric disorders. Figure 6. The DBSNP visualization of different variants covered by the novel exon in MAP7D2 gene. Research plan. I plan to investigate more the detected SNPs in the novel exons/genes and expand the search to the known exonic genes in DBSNP and ClinVar. The Single Nucleotide Polymorphism Database, DBSNP, is a free public archive for genetic variation for humans. The database contains a range of molecular variation that are clinically relevant variations, such as SNPs, short deletion, and insertion polymorphisms, microsatellite markers. By submitting the list of novel exons/genes to DBSNP, we can determine the different variants covered in those regions, the type of those variants, and their clinical significance. I will not restrict my search to neurological diseases only, but I will also use ClinVar to investigate any potential relationships among human variations that I can find in the novel exons/genes and different phenotypes. I also plan to search the Genome-Wide Association Studies GWAS catalog to investigate further the different variants and their related studies. The GWAS catalog also allows visualizations of the variant trait associations, which are mapped onto their chromosomal positions on the human genome. Finally, I also plan to perform co-expression analysis of the novel monoexonic genes that are computationally validated and conduct a correlation analysis between their expression and the detected variants. References. Abascal, F. et al. Loose ends. Almost 1 in 5 human genes still have unresolved coding status. Nucleic acids Res. 46, 7070 to 7084, 2018. Akbarian, S. et al. The PsychEncode Project, Nat. Neurosci. 18, 1707 to 1712, 2015. OKF, Sebastiano V. Offshar PT, Dorothy J.D., Lee L., Williams B.A., Van Backel H., Shat E.E., Rayo Para R.A., Underwood J.G., et al. 2013. Characterization of the human ESC transcriptome by hybrid sequencing. PNAS 110, E4821 E4830. Cassandra Williard, 2018. Expanded Human Gene Tally Reignites Debate, No News in Focus, Nature Genetics Volume 558, p354. Han, S., Liang, Y., Li, Y. and Du, W. Long Non-Coding RNA Identification, Comparing Machine Learning-Based Tools for Long Non-Coding Transcripts Discrimination. Biomed Res. Int. 2016. Feng, Z. et al. Genome Analysis Bayesian Integrative Model for Multiomics Data with Missingness. 34, 3801 to 3808, 2018. Frankish, A. et al. Comparison of gene code and RefSeq gene annotation and the impact of reference genset on variant effect prediction. VMC Genomics 16, 1-11, 2015. Fickett, J. W. Recognition of protein coding regions in DNA sequences. Nucleic acids Res. 10, 5303 to 5318, 1982. Fickett, J. W. and Tung, C. S. 1992. Assessment of protein coding measures. Nucleic acids Res. 20, 6441 to 6450. Frankish, A. et al. Comparison of gene code and RefSeq gene annotation and the impact of reference genset on variant effect prediction. VMC Genomics 16, 1-11, 2015. Ghosh, J., Lee, Y. and Mitra, R. On the use of Cauchy prior distributions. Bayesian Anal. 13, 359 to 383, 2018. Jaraborg N., Bernie E., Durbin R., 1999. Comparative analysis of non-coding regions of 77 orthologous mouse and human gene pairs. Lei Cao, K. A. Martin, P.G., Robert Granier, C. Bessie, P. Sparse Canonical Methods for Biological Data Integration, Application to a Cross-Platform Study. BMC Bioinform. 2009, 10, 34. Levine, M. 1999. What is Bayesian statistics and why everything else is wrong? The Journal of Undergraduate Mathematics and Its Applications, 2165-174. Mudge, J. M. and Harrow, J. The State of Play in Higher Eukaryote Gene Annotation, Nat. Reverend Jennett. 17, 758-772, 2016. Meng C., Zalesnik, O. A., Thallinger, G. G., Custer, B., Golami, A. M., Culhane, A. C. Dimension reduction techniques for the integrative analysis of multi-omics data. Brief, Bioinform. 2016, 17, 628-641. Namuva Oy, Li M., Rychkov Psi, Vlasova N.V., Grigorenko E.L. 2013. Gene Expression in the Human Brain, the Current State of the Study of Specificity and Spatiotemporal Dynamics. Child Dev. 84, 76-88. Perdio, M., et al. Chess, a new human gene catalog curated from thousands of large-scale RNA sequencing experiments, reveals extensive transcriptional noise. 1-14, 2018. Salzburg, S.L. Next Generation Genome Annotation. We still struggle to get it right. Genome Biol. 20, 19-21, 2019. Sine, M., S. and Shu, J. S. J. Bayesian Inference of a Multivariate Regression Model. J. Probab, Stat. 2014, 2014. The ENCODE Project Consortium. 2012. An Integrated Encyclopedia of DNA Elements in the Human Genome. Nature 489, 57-74. Trapnell, C. et al. Differential Gene and Transcript Expression Analysis of RNA-seq Experiments with Top Hat and Cufflinks, Nat Protac. 7, 562-578, 2012. Valderrama Bahamondes, G. I. and Frulish, H. MCMC Techniques for Parameter Estimation of ODA-Based Models in Systems Biology, Front, Oppel, Math, Stat. 5, 1-10, 2019. Van Ravenswaaij I. D., Cassie, P. and Brown, S.D. A Simple Introduction to Markov Chain Monte Carlo Sampling, Psychon, Bull, Rev. 25, 143-154, 2018. Wu, C., Zhou, F., Ren, J., Li, X., and Zhang, Y. A Selective Review of Multi-Level Omics Data Integration Using Variable Selection. 1-25, 2019. DOI. 10.3390/HT8010004. Wang, L., et al. CP8, Coding Potential Assessment Tool Using an Alignment-Free Logistic Regression Model. Nucleic Acids Res. 41, 1-7, 2013. Zhao, S. and Zhang, B. A Comprehensive Evaluation of Ensemble, RefSeq, and UCSC Annotations in the Context of RNA-seq Read Mapping and Gene Quantification. BMC Genomics 16, 1-14, 2015.